Hi, I'm Brandy Sargent, editor of Cell Culture Dish, and joining me today is Dr. Jon Rowley. He's the founder and chief product officer for RoosterBio. Thank you for joining us today.
Anytime, Brandy.
I wanted to start by asking you, what drives the decision for the manufacturing platform for cell therapy manufacturing?
I feel that the biggest thing that drives the decision-making process on what manufacturing platform to use is actually has to be driven by lot size, right? So what you have to do is really understand what your commercially relevant lot size is, and then work backwards from there. And then what that does is it really helps to make sure that the manufacturing platform that you're in is scalable. Now at the same time, you have to balance that with the stage of development. So if you're just getting into phase one, the most important thing about getting into phase one is speed, right? If you're going to fail fast, right? So it's just balancing then the phase of development, final lot size, right? So we spend a lot of our time in mesenchymal stem cells, and these are adherent cells, and so traditionally they've been grown in flasks, and those manufacturing processes are pretty tried and true, and you can very quickly get a 10-layer manufacturing process into a clean room and generate phase one material, and so that speed to your clinical product in phase one is probably the most important thing. And however, 10 layers for almost all indications are not really a commercially relevant source. And so then while you're de-risking your product in your phase one, you will then quickly go down the decision-making route. Our 10 layers going to take you all the way to commercial? If not, when do you implement bioreactors? And then really it's like getting into bioreactors in phase two and phase three is the way to go. But again, it has to be, but bioreactors are not 100% necessary in all indications. And so it's really doing that lot size analysis and balancing it with phase of development.
And to follow up on that, where do you see the manufacturing bottlenecks for cell therapy-based products, and then how do those shift or adjust as a scale-up occurs?
Yeah, no, that's a great question. So my answer is going to be more focused on allogeneic, right? So autologous and allogeneic have very different manufacturing challenges. So, but for allogeneic, or patient-specific, or universal cell, where one patient's worth of cells go and treats hundreds to thousands of patient doses, the manufacturing bottleneck, even at small scales where your lot size is in the billions of cells, is not necessarily in the culture. You could easily double the culture output, but it's really the downstream processing, right? And so if there's anything that we're learning from the field of monoclonal antibody production, is to really try to anticipate what your downstream processing bottlenecks are and proactively develop the technologies to address this. So even just clarification and concentration of your harvest, right? So for mesenchymal stem cells, the cells are harvested in trypsin, right? And you can have tens of liters today, easily hundreds of liters of cells in quenched trypsin that you need to completely get out of that residual manufacturing process components, and the traditional centrifugation just doesn't do that. So there's been some continuous centrifugation technologies that have been developed. They're being tested and implemented in cell manufacturing today that could be scalable to hundreds of liters to eventually thousands of liters. But I would say that the majority of cell therapy processes that I know of currently do not implement that yet. And if you don't get that down, it's really hard to fix later on. So the downstream processing is definitely a challenge today. It's not just in the centrifugation. It's also the formulation, the fill, and even the cryopreservation, right? So cryopreserving thousands of patient doses is not there's not really equipment out there to do that today. And then how you inventory it and move it into inventory without getting intermittent thaws that happen throughout. That whole back end of the scale up process has a lot of work to do. Most people talk about scale up in bioreactors, and I think that's very important to do, and it's hard. But once you're in bioreactors, you don't want to wait another 5 years for the rest of the technology to come into play. And so it's really important for equipment providers to be really working on that, what that downstream looks like.
Right. And to go along with that, how does the manufacturing platform affect the cost of goods for some cell-based therapy products?
Yeah, that's another great question. We spend a lot of time, coaching different therapeutic companies through this, and when you do a 10-layer process, you can actually generate a tremendous chunk of the cost of goods just in labor and media changes, labor in harvesting, because it's kind of a scale out of a small piece. But as you get into bioreactors, something that can take even 10 billion cells, you could take 10 to 15 people to harvest with a tremendous amount of labor, and you probably save 80% of the labor by moving to a bioreactor. And then once you're in bioreactors and you go from say 10 billion to 100 billion to several hundred billion, because everything is moving via pumps, and it's not operators that are moving individual small components. The amount of labor per unit dose manufactured just goes down tremendously, and that is a huge savings. That can save you probably up to 30% of the cost of goods. And then as you get into the bioreactors and get into the media optimization, and then also the quality control testing, which can then be distributed out through many more product doses. I think that as you get your lot sizes up to thousands of patient doses from bioreactor-based processes, that you're definitely looking at having your cost of goods, if not more.
Right. And I want to kind of ask you a little bit of a tough question.
Sure.
What do you think the biggest opportunities are for standardization in regenerative medicine today?
The standardization on the manufacturing technologies is definitely a big opportunity. But specifically focused on mesenchymal stem cells, MSCs, right, where we focus. We think that there's a really tremendous amount of opportunity in removing, for what we see as a significant amount of redundancy throughout the field. So MSCs are being used in lots of different applications, just in regular cell therapies, but then also people are improving upon this traditional MSC that is being used from the gene editing technologies are coming in, and you're getting it's a great platform for gene modified MSCs. The MSCs are platforms for creating exosomes and extracellular vesicles. MSCs are the raw materials in several of the engineered tissues, which are a small component of regenerative medicine today, but I think will be the fastest growing over the next 10 years. And so now what you have is tens of companies today, if not hundreds of companies over the next few years, that all need essentially the same thing, which is vast numbers of therapeutic grade MSCs. And so, what we're doing at RoosterBio is actually trying to standardize that the cell banks that they work from, and then the manufacturing process. So if we can help to get everybody into a standardized manufacturing process, generating the same base cell from which they're either gene editing, collecting exosomes from, collecting gene-edited exosomes, those cells going into engineered tissues, which are a vast number of different types of products, but then standardizing the one way to make the bulk MSC, we think that that's probably going to impact a good 30% of regenerative medicine today, and what we're trying to push forward as a business.
And I wanted to close with a question about how big of an impact do you think that government-funded national manufacturing innovation initiatives will have on the industry?
I think it's going to have a tremendous amount. So, these NMIIs, the National Manufacturing Innovation Institutes, and so there's been two funded by Manufacturing USA. The Nimble and the BioFab USA are also called ARMY. And what they're doing is they're investing in the manufacturing sciences. So if you look at the amount of manufacturing progress that was made in the monoclonal antibody space after the first blockbuster drugs were established in the early 90s, That's where the yields went from 100 milligrams per liter to 1 gram per liter to eventually 10 grams per liter. But that investment in manufacturing of monoclonal antibodies and the biologics didn't really happen until companies were making these billion-dollar drugs. We don't have a billion-dollar drug yet in cell therapy. And so having the US government come in and fund these initiatives to several $100 million, proactively investing in the manufacturing sciences aspect, I think that we will then get to commercially relevant manufacturing processes faster as an entire field. And so I think that they're going to tremendously accelerate the entire field. I think it's a really important thing that the government's doing there.
Thank you for joining us at Boston Biotech Week and on BWB TV. I appreciate your time today.
Sure thing. Anytime, Brandy.
Great, and have a great week.
You too.